Welcome everybody to the third session of the HCA Asia 2021 meeting. We, Quan Nguyen from Australia and Angela Wu from Hong Kong, are pleased to chair this session on single-cell and spatial technologies. We have a stellar lineup of 7 speakers from 5 countries to present the latest experimental and computational methods. After the talk, we will have a panel discussion about future development and collaboration in the region. For the online audience, may I remind you that you can type the question in the Q&A questions sections, and the speakers will have time to address your questions. Now, to start, we are delighted to introduce Dr. Shalin H, the head of the single-cell system biology in the immunology division at the Walter and Elijah Hall Institute, to present a plenary talk for this session. And Shalin has been leading the very successful and elegant cellular barcoding technology suite, and we're looking forward to his great talk. All about you, Shalin.
Thanks very much, Quan. Really appreciate the opportunity to speak today. So thank you for inviting me. And indeed, I'd love to talk to you a little bit about the cellular barcoding technologies we're using in the lab for the study of various systems. First and foremost, close to my heart is the study of haematopoiesis, but we're also interested in the study of development and cancer. So, you know, this image here is actually some barcoding data from a breast cancer barcoding study we did, where every column is a piece of a 3-dimensional tumor, and the size of the dot is proportionate to the size of the clone in that tumor. And as you can see across the different pieces in the tumor, there's different clones that are of different sizes. And not only is it beautiful, but it's actually informative to tell you about the 3D structure of the tumor. Now, of course, technologies like spatial technologies are giving us even more granularity, but this was the first really clonal level information we could get. And so what we study is clonal heterogeneity. So imagine a group of 10 cells that you're studying in the lab. They could be stem cells, they could be immune cells. And if you put those 10 cells in culture and they give rise to red, blue, and green cells, there are actually an infinite number of ways 10 cells can give rise to that number of blue, green, and red cells. Here are some examples. You could have 10 cells that are equipotent, and they all give rise to 3 cells each of the colored cells. Alternatively, a subset produce only red, a subset blue, and a subset green. Or alternatively, there's a degree of heterogeneity in fate, not only in what they make, but how many they make. And of course, you add the dimension of time to that, and you've really expanded the possibilities of what a single cell can make. So how can we assess the output of hundreds of single cells in vivo? Because really that's where we want to measure our output ideally. So you could inject a single cell into a single mouse and do that for 100 mice, and you'd have 100 cells. Or alternatively, you could label 100 cells differentially and inject them into the one mouse. So of course, for the researcher and for the mice, the second option is more preferable, and that's what we do. So our first iteration of barcoding that I co-developed in Ton Schumacher's lab in Amsterdam takes a lentivirus where we had 2,500 different DNA barcodes, 98 nucleotide stretches of DNA that we infect into our progenitors of interest at a low MOI, so that there's only, in most cases, 1 integration per cell. So we do it about 10 to 15% transduction efficiency. Then we inject those stem and progenitor cells into an irradiated recipient, and then they divide and differentiate as they ordinarily would. However, this time when they do that, because the barcode is integrated into the DNA with the lentivirus, all the progeny then also inherit that barcode. So that at the end of the experiment, you can take cell type A, take cell type B, and then lyse those cells, do a PCR using a common set of primers for the barcodes, and count them using next-gen sequencing. So here we've got, hang on, let me just put my laser on. Barcode 1 had 2 reads in cell type A, but none in cell type B. That is indicative of a mono outcome progenitor. So this blue progenitor only made cell type A. Barcode 3 gave reads only in cell type B. So it was a mono outcome progenitor only for cell type B. whereas this yellow cell had reads in both cell type A and cell type B, and therefore it was a bi-outcome progenitor. And so in this way, long story short, we identified that for the aficionados, it was lymphoid-primed multipotent progenitor within haematopoiesis, which at that time was considered the last multipotent progenitor in that pathway. And what we found was they are anything but multipotent. And in fact, very few of the progenitors, less than 5%, generated all lineages, and the rest were biased towards either B cell development or dendritic cell development or myeloid cell development or combinations of the two. And so we published that a while ago. And at the end of that study, we knew what heterogeneity looked like. But what we didn't know was what are the molecular programs that control that heterogeneity. And so we sort of faced this Heisenbergian problem. So for those of you who remember the Heisenberg uncertainty principle from chemistry, you can't know a molecular particle's location and its momentum at the same time because measurement of one precludes measurement of the other. So we sort of had the same problem in single cell biology. Imagine a cell that is programmed, destined to make only orange cells. So it divides, and at the end of the culture, you find out, or in vivo experiment, you found out it made orange cells. What you would love to do is to know what was the molecular program of that parental cell that contributed to that, but you don't have that opportunity anymore because they've divided along the way. Alternatively, you could take that cell and put it in a single cell RNA-seq experiment. And that's great. You find out the genomic profile, but apart from, live-seq and perhaps some other technology in the future, you have to destroy the cell to get the full understanding of the molecular makeup of that cell. So you can't get both. And so there are two solutions that you could think of to try and get this. You could either get a time machine and find out the clone that made orange cells, go back in time, assess that for gene expression, find out the gene expression correlates there, and not do that once, maybe go back in time twice just to get good technical replicates, and then do that for 100 or 1,000 different clones. And then you'll be able to pair genes of the mother cell with the fate of the daughter cells. And you should be able to disentangle it that way. In the absence of a time machine, we figured, well, what if we give them a couple of divisions first and then assess some cells for fate and some for RNA-seq? And so that's what we did and came up with this approach called scONE-seq. Now, this is very similar to approaches that Sam Morris, Alon Klein, and others have made where you can pair gene expression with fate using barcoding. In this particular experiment in paper, we didn't use barcoding, but now we're using that with new lentiviruses. But in essence, you expand that clone. In our case, we're studying the output of stem and progenitor cells towards dendritic cell development. We let them expand a couple of days. Then we have some for fate analysis that we assess by flow cytometry at the end of 8 days, and others for gene expression. And we call this clone splitting in sister conditions for parallel fate and RNA-seq, or scONE-seq. And a long story short, in collaboration with Matt Ritchie and Lu Yi Tian, who is a joint PhD student with us, we found those genes that correlate with fate. So normally with single cell RNA-seq, you're looking for highly variable gene expression, cluster marker gene analysis, and then differential expression. But in fact, these genes that I'm showing you on this graph would not have been discovered that way. In fact, only 10% of them overlapped with the traditional means of gene expression, high variable analysis. Instead, these are genes that correlate with clonal fate, which you could not have got without that clone pairing experiment. And in that way, I won't go into the data today. This is just a summary of that. We found 30 genes, 30 new genes, which when we knocked them out, affected the development of the different dendritic cell subtypes. And so not only did we identify them, but we then knocked them out to validate them, because I think that's very important. And we believe this is a really great way to find the molecular regulators of clonal fate. And so we can apply this now to anything, whether it's which iPSCs program very well, which T cell grows a lot in order to contribute to immune response, and why. All right, so I'm going to finish there. You can catch up with this work in immunity, which for which we got the cover by my friend Dario Nucci, who, using an MC Escher inspired artwork, created this stem cell turning into a dendritic cell here. So what I just mentioned has been our workhorse for a number of years, but it's not ideal because you have to take a cell out of its context. You have to label it with lentivirus and then inject it back in. And in the case of haematopoiesis, you have to irradiate that mouse. But wouldn't it be great if we didn't have to irradiate? What if we could barcode cells live while they're inside the animal without ever touching the animal? Wouldn't that be cool? So we came up with barcoding, LOX code barcoding. And this is really the work of Tom Weber, who's a theoretical physicist by training, who came up with this idea. And then without having ever touched a pipette before, I said, come on to Australia and make it. And along with Denise Miles, he did. And so I'm really happy to share this unpublished data with you today. So the LOX code mouse has 13 short DNA barcodes flanked by alternating LOX-P sites. And for those of you who don't know, you can add a Cre recombinase. It's an enzyme. It finds those LOX-P sites and it either flips what's between them or it cuts it out depending on the orientation of the LOX-P sites. And in that way, would you believe this 13 barcode cassette can generate over 30 billion different barcodes? I still don't believe it, but Tom assures me that that's the case. Unfortunately, the diversity is not equal throughout the different lengths of LOX codes, and these most of the 30 billion are extremely rare to be generated, so they're not generated equal frequency. But it doesn't matter because there's plenty of diversity in the lower complexity, more frequent barcodes. And you have to exclude the really common barcodes like a single barcode. Because that happens too frequently, we have to computationally exclude it. But anyway, I digress. So I'm just going to close these windows so I can see my window. And as you can see from this PCR on the right here, after you add tamoxifen, you get recombination of this cassette and you get these different sized elements that you can see here on the left are reflected in the PCR here on the right. And we made a mouse from it. It took us 1.5 years to clone this thing. It was a real pain because the bacteria kept kicking out all these LOX-P repeats, but we got there. And you can think of it like a Rubik's Cube, except a Rubik's Cube doesn't shrink as you turn it, but it does generate all these random possibilities. And you might have also heard of a mouse called Polylox, which was developed by Hans Reimer Rodewald in Germany, I believe. And so that's a fantastic mouse and really pushed the limits on what you could do with LOX-P based barcoding. And in their case, they can generate 1.8 million barcodes. You have to inject tamoxifen, 2.5 mgs, and you currently have to use PacBio sequencing because the LOX code is quite long. In our case, we can generate 30 billion barcodes, so it's 16,000 times more. We can use a 50 times lower dose of tamoxifen, which can be important for certain tamoxifen-mediated toxicity-based Cres. And the great thing is we can use Illumina-MiSeq to get that data. And in fact, we're starting to now also get data on NextSeq. So you can put in a lot more samples, get a lot more throughput with Illumina than you can by PacBio. So that's been a great benefit to us. So one little example here of what we've been working on. We've working on several projects, but one is mouse embryogenesis. And this was led by Tom in collaboration with Christine Bibbin, a very talented developmental biologist in Simia Taudi's lab. So during embryogenesis, you have that one cell that at embryonic day 5.5 in the mouse turns into an epiblast, which is essentially a cup or a ball of 200 cells. And there's been some debate whether those 200 cells contribute equally to the end embryo or whether some of those cells are already biased towards one tissue or the other. And by embryonic day 12.5, you've now got an embryo which has got most of the major organs, limb buds, brain developed, etc. So what we did was cross a male LOX code mouse to a female Rosa Cree ERT2 mouse and plug the female, and then at 5.5 days later, we would inject tamoxifen, and then 7 days later, harvest that embryo. This is a little tiny cell with about 10 million cells, and Christine then chops up that embryo for bulk barcode PCR into 40 pieces. It's a little tiny corn kernel size, or grain of rice size thing. It's amazing. We did either bulk PCR or we also did single-cell RNA-seq. And other embryos, we took a cross-section for spatial RNA-seq analysis with Visium. And so I won't go into all of the tissues that we harvested, but here's a rough survey of the different tissues we cut from the embryo to do LOX code analysis. And then we can ask the question of that 200 cell stage, of those 200 cells, did they all make all tissues equally, or were there some biased for some tissues and others biased for others? So with a PCR-based readout where you take a whole tissue, you lyse the tissue as a whole, you PCR up the barcodes and find out what's the composition is, similar to what I showed you with the lentivirus barcoding. This is the results we found. So you've got the LOX codes along the x-axis here. So each column is one LOX code. It's one cell in that epiblast that contributed to lineage. And then in rows, we have all of the different parts of that 12.5-day embryo. And as you can see, if every cell contributed to everything, you just see a sea of yellow there. You'd see everything everywhere. But we don't see that. We see some clones with different biases. And just to highlight a couple of them here, I'll have to do it by hand. So on the left, you see most of the multipotent clones. They're contributing really to all of the different tissues here. But now you can see pockets of clones from multiple different embryos, independent embryos, that contributed mostly to the limbs, right? The left limb and the hind limbs and the forelimbs, and not much else. Or a lot of these clones that contributed to neural tissue and not much else. So we're seeing quite some patterns here. It's not as black and white as we found with the hematopoietic progenitors, but there is quite a bit of bias in what those clones make. And so here's a couple of examples of those clones. So here's what we call a butterfly plot of that embryo. Here's the identity of the LOX code that we're looking at, -1, -8, -13. And the color here is representative of the clonal contribution of that clone, as specified in this column here for number 1, to each of these tissues. So as you can see, this clone was really biased towards contribution to some of these internal organs and less so to the neural crest and brain. This clone here was very biased towards generating the posterior of the mouse rather than the anterior, like the brain and the face and the neural crest. And this one was very internal organ biased, we found. So it didn't generate any of the neurons or neural crest, mostly just internal organs here. So you get all these different flavors of clonal bias. And to give you an example about the reproducibility, here's the aggregate of 3 mice worth of clones contributing to the brain tissue here, midbrain, hindbrain, forebrain, and the limbs, hind limb, forelimb, etc. And as you can see, there's a correlation in developmental origin with the limbs that is separate to the developmental origin of the brain, which is very cool. And to demonstrate reproducibility, sorry, my screen, oh, there it is. You can see here that 3 independent mice all showed a very similar fate in that way. Okay. Are you seeing the data slide or is everything okay on your end, Kwan?
We're seeing the heat map.
You're seeing just the heat map. Okay. All right. I've got this screen covering that. But anyway, as long as you can see that, that's what's important. I'll try and move this. Oh, there we go. Get out of the way. All right, you'll just have to tell me when there's a couple of minutes left, Kwan, because I've had to move the screen. The other way we can project that tissue is on a UMAP. And so here, each dot represents a part of that tissue, and the UMAP is representative of how the different clones contributed to those tissues and laid it out. And you can see the neural tissue is enriched in one area, the mesenchyme in the other area, and we even see a bit of anterior-posterior axis going on here. I don't have time at the moment, but what does this mean in terms of the embryo? Were those cells already programmed to make these tissues, or was it an artifact of the location they were in as the embryo started to develop? I don't think we'll ever be able to answer that question. But nevertheless, this was the destiny of the clones, and it is a pattern that is reproducible. And lastly, second last, I want to talk about spatial RNA-seq. So we took that embryo, and with the help of Casey from the single cell core, we had a look at it. And this is now this is the day 12 embryo here. Each dot represents a RNA capture spot, and we've clustered them and then annotated them based on their gene expression values. Christine's done this manually. And here's a UMAP projection of each of those clusters, as you can see. And so you can see that there is a sort of a UMAP axis that goes down here from the hindbrain down to the forebrain, as you can see here, and it kind of goes in that order here. And then if you look at the back of the mouse, you've got the, again, the posterior of the mouse all the way, of the embryo all the way down here, which is the anterior. And so there's some structure to this data that sort of recapitulates the 3D geography of that embryo. But now we can ask, what about the LOX codes that were detected in those patches? So again, here are the, and now we've just done this in alphabetical order. We need to actually try and cluster this a bit better. But as you can see, the different clones can be detected in each of these tissues, and some are genuinely multipotent, yes, and others appear to be enriched in some tissues and not others. And so the way we can visualize that in another way is in these, we call them meta-patch enrichment plots. So basically, each patch represents a transcriptional identity, and then the color of that patch is represented by the number of times we detected a LOX code transcript in that area. So it's not many at this point in time, but we can see now in a spatial context the clonal contribution to each part of the embryo. That's one example of one LOX code up here, and here's another example of another one here. And as you can see, this one was as close to multipotent as we could get, and this one seemed to be enriched for the internal organs and the face, but not so much for the neural tissue, which is quite interesting. All right, and finally with single-cell RNA-seq, so Christine chopped up those 40 tissues, digested them, we multi-seq hashtagged all of them, and then ran them all on 10x. So we've got 40 tissues separately hashtagged worth of single-cell RNA-seq information. You can look at the annotation here, but roughly speaking, those are the neural tissues on the left. It's the limbs and the mesenchyme at the bottom, and all the different organs there at the top. And so, as you know, with current Visium, you're working with a patch of 50 to 100 cells. So it could be that there's a clone that, well, there's a patch that's, you know, a bit of mesenchyme and a bit of neuron that's in the same patch, and then you don't really know, was that barcode detected in one or the other or both? With single cell, you can. And so, this is another way we're annotating the clusters now based on their gene expression values. And I'll just give you two example clones here. This clone 1 to 11 was really enriched towards contributing to neural tissue, but not the limbs, the mesenchyme, and most of the internal organs. It contributed a little bit of this one and a little bit of that one. Whereas on the other hand, this was a really mesenchyme biased clone. It contributed to all of the different organs and limbs, but not that much to the neural tissue. So now we've got this beautiful capacity to track a in vivo derived, in situ derived barcode and where it contributes to tissues using bulk, readout, spatial RNA-seq, and single cell. So with that, I want to wrap up and thank all of the members of the team, but in particular the stories I mentioned today. The scIS-seq story was driven by Lu Yi-Tian, Sarah Tamai, and in collaboration with Matt Ritchie. And the data I just presented now was driven mostly by Tom Weber, Christine Bibbin from Sam Towdy's lab, and now continuing with Sarah Tamai in the study of haematopoiesis. And with that, I'd like to thank you for your attention.
Thank you very much, Shalin. We still have plenty of time for questions. So I may start first with a question. When you induce the tamoxifen for the in situ barcoding, what the effect of the dosage of the tamoxifen you think that it may have on the the diversity of the barcodes and the efficiency for the barcoding combination?
What was the variable you asked about? The time of
so
injection?
the dosage of the tamoxifen when you apply it to the mouse at day 5.5, right? The embryo at day 5.5. If you increase the dosage of tamoxifen, would that increase the diversity of the lock code? Yeah.
No, so with LOX codes, you have this sweet spot. So at the extreme, if you don't recombine it, you have 1 barcode. At the end of, if you recombine it to the end, you have 14 barcodes because it's just 13 times 2, 26 barcodes because they've just recombined to the end. So you've got 1 and 26, and in between is where you have your diversity. So we have to find a sweet spot. Basically, we do different injections in our model of interest and then find out which one gives the best ladder. So like I showed in that PCR, you want to have the best ladder, and that's hopefully going to give you the it will give you the maximum diversity that the mouse allows you to have.
That's great, thanks. We have a question from Christine. Are you confident that the LOX code recombination is happening synchronously and not later than you thought in some cells? Could it be the cell cycle linked?
Yeah, so I don't know about cell cycle per se, but what I can say is that at least in vitro, when we do a short pulse, like an hour pulse and then wash it out, it's pretty fixed. So you get recombination, I think, within a couple of hours, and then it doesn't change over time. But of course, that's in vitro. And in vivo in a mouse, you can't just wash out the mouse very easily. So you're relying on a certain amount of kinetics. So how long that recombination goes on, especially in this question of embryonic development, Christine, I think, has done some time course experiments where she injects and then harvests the embryo 1 hour, a few hours later, and so on. And we're currently waiting on that data. So we don't know the answer to that, but presumably it's not going to happen just once and then stop it. There's probably going to be a little bit of a hangover, whether it's cell cycle related or not. We could probably test that in vitro. We haven't done that. Other people might already know the answer to this.
Thank you. Another question is from Sham. He asked, if a clone gives rise to only limb, does that necessarily mean it was limb biased?
So here it comes down to semantics and terminology. Yes, it was limb biased, but what made it limb biased? Was it an artifact of where it ended up? Like, imagine you get stuck in a mosh pit and that part of the mosh pit like takes you toward the exit. You weren't predestined to leave. You would just happen to be at that part of the mosh pit at the start of it. Versus, and I think what you're really getting at is, was it predestined to make the limbs based on where it was? Like I said during my talk, I don't think it's going to be easy to answer, at least not with this technology. And I think it's going to be a case of if one, not us, if someone were to find genes that predilected a single cell to make limbs and you did some sort of labeling experiment, then you might be able to kind of validate that it was predestined based on molecular programming rather than luck.
Yeah, and Sham has a theory about that. It could be stochastic. So if some daughter cells survive and then give the rise to the limb.
Yes, that's true. So one thing I should mention is that classic embryology experiment, I'm not an embryologist by any stretch of the imagination, but classic experiments have taken a cell from one part, put it in another, and it turns out that the cell adopts the fate of the environment you put it in, not where it came from. So there's a high degree of plasticity still at the time of labeling. Not to say there's not a little bit of imprinting, but certainly it is highly plastic.
Great, thank you very much, Shalin. For the interest of time, we'll move to the next speaker.
Thank you.
Thank you. So I'm very happy to introduce Angela Wu, who is my co-chair for this session. And Angela is from the Division of Life Science and the Department of Chemical and Biological Engineering at the Hong Kong University of Science and Technology. Angela Wu, over to you.
Thanks, Quan. All right, let me share my slides first. Can you guys all see and hear okay?
Yes.
All right, thanks, Quan. Okay, so hi everyone, my name is Angela, and thanks, Quan, for the introduction. I guess I'll just jump straight into my talk. Today I'm going to tell you about a novel single-cell multi-omics approach that we've invented recently called single-cell one-seq. And this is for simultaneous whole genome sequencing and RNA sequencing from single cells, and it's also compatible with frozen tissue. So we really would like, to make a method that is compatible for frozen clinical samples, and that's sort of the, the starting point of this technology development. So I think for this audience, I don't need to talk too much about, why single-cell multi-omics is informative, but I think in particular, it's very informative for understanding cancer. I mean, as you know, there are many layers of regulation between the genome, which is, the blueprint, and the transcriptome, which is sort of the proxy for the phenotype. And so if you can simultaneously see multiple layers of information, that can be very informative, especially for cancer, where in the bulk, it's already been demonstrated that cancers can be highly heterogeneous at multiple levels. And I think some of our other speakers will speak to this as well. So that's sort of our motivation for creating a multi-omic DNA plus RNA method. And definitely there were some pioneers in this field. I think one of my following speakers is one of the earlier inventors of this, of one of the approaches. But in general, those earlier methods use either a separation strategy or a split strategy. But to sort of save on time, I'll just kind of summarize that they all sort of have their different pros and cons, but most of them use a poly-A approach or have to, separate the RNA from the DNA separately, which one makes it kind of difficult to scale. And two, it can sort of preclude certain types of analyses like total RNA sequencing. So we wanted to have a method that was more sort of all-encompassing and versatile. And in particular, as I mentioned, could work with clinical samples, which tend to exist in frozen sort of formats in the biobank. So our method, the goal is to have a one-pot reaction where you put the cell into one tube or one container, and then you just keep adding stuff to it without having to do too much processing in between. So our method doesn't require any centrifugation or beads, no sample splitting, and it doesn't require microfluidics, just your standard molecular biology lab equipment. And it can be automated to use a liquid handler, which is something that we have recently done. And also it fits with a standard FACS workflow up front. So if you wanted to do sorted cells or enriched populations, that's also fine too. And the cost of this is about $2 per cell, which is similar to doing SMART-seq. Your mileage may obviously vary depending on the sequencing platform that you're using and cost of your reagents. Generally speaking, the way that our approach works is that for tagging the DNA part of the cell, that is the whole genome sequencing, we use TN5 to add a DNA-specific barcode and a co-amplification primer. And then to capture the RNA, we do an RT with an RNA-specific barcode and the same co-amplification primer. So after you do all the molecular biology and processing, at the end in your pre-amplified pot, what you should have is the fragments of whole genome or fragments of the genome DNA with a DNA-specific barcode and an amplification primer, and then also the RNA in the same pool, which is the cDNA now, with an RNA-specific barcode and also the same amplification primer. So then we just do co-amplification of everything together, sequence it, and then in silico, we can separate which fragments arose from the genome and which fragments from the transcriptome. So then we just, first to verify that it was technically working well, we did a comparison between our method and SMART-seq on the RNA side, and then compared with bulk for the DNA side. So on the left here, I'm showing the sensitivity of the method as compared to SMART-seq 2. You can see here, this is our method and this is SMART-seq 2. And in terms of the gene detection number, we're doing a little bit better than SMART-seq 2. This is only because, as I mentioned, our method is a total RNA sequencing method and also a full-length RNA sequencing method. So it's not like 10x. It's not a 3'. It's not a 3' method, and we can capture also the non-polyadenylated sequences as well. So that's probably why we're capturing a higher gene number. And in terms of the gene body coverage compared to SMART-seq 2, it's also very, very comparable in terms of covering the entire gene body. So we were pretty happy that the transcriptome part was working well. We also have other evidence from cell lines and PBMC, which I don't have time to show today, but please just, you can take a look at our preprint later if you'd like to know more. So on the DNA side, we also checked whether we could detect copy number changes and alterations just like other single cell whole genome sequencing methods. So for, here we're looking at HCT116 cells, which are relatively homogeneous. And this is the true bulk sequencing data from HCT116, where you can see the amplifications and deletions from the genome. Here is the pseudo bulk that we reconstructed from 96 of the cells sequenced using our method. And you can see that, again, we capture the same copy number changes right here. And then with our single cell one-seq method, which I may remind you that this genome data comes from the same set of cells as this RNA-seq data. This is just a randomly picked single cell. And again, we can call the copy number changes from the single cell as well. And it matches to the bulk and the pseudo bulk. So we also did a brief benchmarking compared to these previous methods that were published. And, those methods were published quite some time ago. So, I think our method really benefits from being a one-tube approach where we sort of minimize the loss. And you can see that from the DNA data in particular. In this Lorenz curve, which illustrates the uniformity of coverage for single-cell whole genome sequencing data, these gray dotted lines are from the previously published methods where people sequence RNA and DNA together from the same cell. And the purple line is our method, which is, again, randomly picked single cells. And you can see that this is closer to the ideal and also the bulk, which is shown in light blue here. So the closer this line is to the unity line, the better the uniformity of coverage across the genome. And same with sort of the overall consistency of coverage. This is sort of a measure of reproducibility on the DNA side, and you can see that our method has a much lower coefficient of variation, which means that it's more consistent. On the RNA side, you can see that we, again, look at the sensitivity and also sort of the linearity of the method using ERCC controls spiked into the cell sample. And you can see that our profile for single cell one-seq is very comparable to SMART-seq2, both in terms of the sensitivity and also the linearity of the measurement. Whereas the previous methods slightly lose out on the sensitivity a little bit, I think, again, because of our method not having to separate out any materials. And so we sort of minimize the loss from the sample. So just sort of as a brief summary of our method compared to some of the other comparable methods, we have a much shorter experimental time, just 8 hours for one single plate. Of course, if you're using a liquid handler, this can be scaled up quite a bit more within the same or roughly similar time frame. There's only one purification step in our protocol versus 2 or 3 for the others. And our amplification success rate is 90%, which is quite a bit higher. And again, I think it's because we don't need to transfer samples and stuff. And again, our method is the only one that can be used for total RNA-seq. So I think that can be quite advantageous for certain biological questions. Okay, so, and just to briefly mention that for total RNA-seq, we do also have a single cell ribosomal or mitochondrial sequence depletion method called single cell DASH, and that's already been published, so you can look at that. And then very quickly to tell you about how we've applied this method in the clinical setting. So we collaborated with clinicians at the Chinese University of Hong Kong and also a collaborator at HCA to study 2-year frozen grade 4 astrocytoma sample from a patient. Gliomas are actually very complex in terms of their genomic heterogeneity. You can see on the left here, this is just a flow chart of what clinicians have to look at in terms of the genomic heterogeneity of this type of disease or cancer. And on the right, this is an illustration of the transcriptomic heterogeneity of gliomas. So they can be very heterogeneous in terms of their transcriptional profile as well. So that's why we thought this would be an interesting method to use to look at gliomas. Plus, they have a very poor survival as well. So for the patient that we looked at, this was an interesting patient. She had a primary tumor and then underwent surgery actually 2 more times because she had 2 different recurrences of her astrocytoma. And for the primary and the first recurrent tumor, we could only get bulk data. So we had whole exome sequencing and RNA-seq. And also for the second recurrent tumor, we had the whole exome, the 10x single nucleus data, and then the single cell one-seq data from us. And we generated about 1,200 single cell nuclei from using our method and constructed both the clonal analysis and also the transcriptomic cell type analysis for this patient. So you can see here from the DNA data that we got using our one-seq method, we could identify many of the common mutations or deletions that occur in glioblastomas, or sorry, astrocytomas, they're now called. But most importantly, we found that in this patient, there were 3 different subclones of the tumor. And you can see from the sort of proportion here that there's one huge major clone right here, which we call clone 3. And then clone 2 is smaller and clone 1 is even a little bit smaller. And the difference between clone 2 and clone 3 from the profile you can see is more similar, whereas clone 1 is quite a bit different. And actually, based on the sort of lineage map that we drew, using the copy number alterations that we identified, this second recurrent clone 1 is actually more similar to the primary clone coming from the patient, as in the very first occurrence of this tumor. Whereas the second
1
recurrence,
minute left.
Yes. Whereas compared to clone 2 and clone 3 from the second recurrence, those are actually very much different from the primary clone. So this may suggest something about the progression of the tumor. And then when we look at the transcriptomic profile using our friendly UMAP, you can see here we have a lot of different normal cell populations right here on the outskirts. In the center, this giant blob is the tumor clone. And actually, currently, it's labeled by the transcriptomic subtype of the tumor. But when we overlay it with the DNA clone information, so this is still the RNA-based clustering UMAP, but the colors are now showing the DNA clonal information. You can see that actually clone 1, the one that looks a lot like the primary tumor, it actually falls into the normal cell population, or based on the RNA, they look normal, and they look like astrocytes. So when we compare this tumor, normal-like tumor, with the actual normal astrocytes, we get some upregulated genes like GRIA1 and ADCY8. And GRIA1 is particularly interesting because we found that actually this is a, yes, give me 1 minute and I'll be done with this slide. Okay, so with the GRIA1 gene, it's very interesting because it is the only one of these AMPAR receptors that can form a homodimer and then create a sort of always-on calcium channel that can basically have a synapse be on all the time. And so remember that this is a brain tumor. And so we suspect that this clone 1, this very unique clone 1 that looks very much like an astrocyte, might play an important role in the tumor to neuron communication in the microenvironment. Okay, so I'm going to skip ahead and just briefly tell you that we also have worked on a mouse lemur single cell atlas, which you can look forward to the preprint coming out very, very soon. And we also have a data integration algorithm called Portal that, again, also the preprint will be coming very, very soon. And so, again, these are my acknowledgements. This is my group. Special thanks to actually Jianbin, who's another speaker, and Yanyi for their support in this project, and also, of course, to my collaborators and funders. And I will be very happy now to take any questions.
Yeah, great talk. So we have quite many questions. You may take time to answer them later. But for now, I may just ask some quick questions
Sure.
from the audience. So the first one is the HCA-1-seq data set publicly available?
So not yet, because it's under review right now. So the data currently is just available to reviewers. But once it's published, I mean, you can definitely contact us. And if there's opportunities for collaboration, we'd be happy to make both the protocol and also the data available to you.
Great. And the second question is, can you call SNVs as well?
Yeah, we get that question a lot. I think currently, some of it might be related to sequencing depth, as it might be familiar to many people. So currently, we're sequencing at about 1 million reads per cell, and that's for both the DNA and the RNA. And I think if we sequence a little bit deeper, we can probably get some SNVs with a reasonable level of confidence. But currently, I think it's difficult to call them confidently. And again, I think it's partly because of the coverage and the sequencing depth. So yeah, that's something I'd like to explore more.
Okay, great. And another question is about one of the slides. Is clone 1 malignant or pre-malignant?
Very, very good question. So I think it's we couldn't really prove that from this particular data set because it's just one patient, and also the patient has passed away, and so we can't collect more samples, and we can't really do any functional studies on this sample anymore either, because we couldn't, generate a cell line or anything. But I think we have some interesting hypotheses about clone 1, and so the next step is for us to profile additional patients and see if this type of clone 1 with these, similar features are also present in many other glioma patients? And if so, then it would be very interesting to try to do some functional studies on that in either mice or other types of cell models.
Thanks, Angela. There's another question from Chung. You may answer that question later.
I will. Thank you.
For the interest. Thanks, Angela. Let's move to our next speaker. I'd like to introduce Jianbin Wang. And Jianbin is assistant professor of life science at Tsinghua University in Beijing. And Jianbin gonna talk about the technologies for studying immunity development, single cell analysis, and beyond. Please, Jianbin.
All right, thank you, Glenn. Great honor to be here. So this is Jianbin from Tsinghua University, and my lab is a technology-oriented lab. So we spend a lot of time developing methods for scientists and doctors. So and also we'd like to get some inspiration through this kind of collaboration to be more or better technologies and to solve the problems of biomedical research and also clinical diagnosis and treatment. And today we are going to talk about some research about the development of plasma cells. And to begin with, here's the brief result of a recent study we just recently got accepted. We cannot see each other because of the pandemic, and many groups are trying to get powerful antibodies for treatment and to understand how we can fight against the virus. So this is a little bit of effort we tried to search for the antibodies from the patient recovered from the disease, and we use flow cytometry to get the single cells against the RBD of the virus. And for each single cell, we clone and the antibody, and then try to see how good the antibody works. And this is the routine strategy we use for across the world today to get the antibody against the virus. Of course, it works, but from the technology point of view, it's not the perfect. First of all, we can see that the method uses memory B cells, which is not the antibody producing cells. And for most of the, because plasma cells do not have this receptor on the cell surface for your purification, and we only use memory B cells. And so this is the blood, and you can only use the cells through RNA-seq and cloning to do ELISA. On the other side, we do have antibody molecules in the plasma, but we can only do ELISA to get the general idea how well, how strong the serum is. So from our point of view, we want to solve the problem through the technology development to see whether we can do better to understand how the plasma cell works, and also whether we can directly use the antibody molecules from the cells, from the blood to do better work. So to do this, we collaborate with Dr. Qi Hai from Tsinghua University's Institute of Immunology, and we use a mouse model. And the mouse model is a reporter of the BLIMP1, which is the driver of the plasma cells. We immunize the cells with antigen and then wait to a different time and get the cells from the spleen and the bone marrow and enrich the cells and then to perform single cell RNA-seq. And this is a very routine analysis. We have, although they are highly pure, uniformed plasma cells, we can still see different distribution of the transcriptome profile, and also the different groups of cells at different time at different organs, for example, bone marrow and spleen. And some of the cell groups show a biased representation in either the bone marrow or the spleen, also across a time spot. And to further understand the cell heterogeneity, we perform B cell receptor sequencing and assembly. Then we can see that there's a biased representation of the isotype of the antibody across the population, and some of And most of the cells express IgM, and some of them also express IgA or IgG. And also we can see that most of the antibodies that mature through hypermutation, but some, for example, C1, C5, and C8 and C7 have very little mutation accumulated. And because this is a mouse model, we know we have prior knowledge about the antibody and the antigen. So we map the cells bearing the specific antibody against the antigen to this distribution. And we can see that most of the antibody specific to the antigen reside in cells cluster 8 and 9, and also their abundance change for time and different time and also the organ. And through these quantitative analysis, we can see that only in bone marrow and in C8 that the high affinity antibody accumulate through time, and in other time or organ, the high affinity antibody either not existed or declined through time. So we can claim that the cell 8, cluster 8 cells in the bone marrow represent the long-lived plasma cells, which we believe are the key players in maintaining long-term immunity against viral infection. And going back to the transcriptomics data, we can see that we can get gene markers for the different cell clusters, and we use the markers to flow sorted the long-lived plasma cell that we want. And also we can confirm that only in bone marrow that after the 2 weeks of acute immunization, that the specific group of cells through these 2 markers, we can get more accumulated cell representation. And also we notice that we can notice that besides cluster 8, cluster 7 also has the similar gene structure profile, and cluster 7 has a very large clone structure. And also we see that from different time points, from different mice, they share the exact same B cell receptor sequence across different independent mice, suggesting that cluster 7 has a very strong polyclone structure across mice, the population. And we use barcoding to get B cells from 6 independent mice. Okay, after the integration, we can see that the gene expression profile is very uniform. And for each of the 6 mice, we see that they share at least some kind of clones with other mouse, suggesting that this cluster represents a very special kind of long-lived plasma cells, and also it's shared not only persisted through time, but also shared by different mice. And we use the DT, the toxin, to kill all the cells through this BLIMP1 driven receptor expression. And we can see that after the treatment of the toxin, the plasma cell of the C7 got eliminated, but after through time, the cells recovered, suggesting that the cells may recognize either the environment or the self-antigen, autoantigen from the mice, which can be regenerated. And also, before and after the treatment, we can see that the cell recover from this within the group has very reproducible and stable gene expression profile, suggesting that they are long-lived plasma cells recognizing some persisting antigens within the body. Okay, that's the part we try to uncover the long-lived plasma cells from the cellular proportion of the blood, but we also said that we want to know what kind of more knowledge we can extract from the molecule or the protein side. So we try to apply different kind of biochemistry method to study the molecules. For example, we can use antibody-based analysis to get the antigen from the blood. For example, we have the antibody capturing the antigen, and for example, we are using GIP and anti-GIF antibodies. We can use mass spec to analyze the complex protein and under the physiological condition, and we can also specifically separate different mutations within the antigen. For example, 2 different mutant of the GIP, which are only one amino acid different from each other. And also we can use antigen-based antibody analysis. For example, we have the RBD. We try to this is the molecular weight of the RBD pure proteins. We have three FAB site FAB fragments from three different antibodies, and that's the pure the molecular weight of the pure antibody. And we combine them together, and we can see that each complex represents a different profile under the mass spec. And after we dissociate the complex, we can reproducibly analyze the RBD antigen and also the FAB site fragment of the antibody. So with this method, we can combine the mass spec analysis with the single cell data. We have single cell data. We have the BCR sequence, so we can calculate the theoretical molecular weight of the antibody. Then we have the plasma. We have the mass spec, so we can separate the complex using the molecular weight, and we can see which one is stronger, which one is weaker, and through this molecular weight, we can we can correlate the mass spec data to the single cell data in order to understand what kind of molecule, what kind of biochemistry properties we can get from the plasma, and what kind of cells it is for single cell BCR and single cell transcriptomic analysis. With this, we think we can better propel the research of antibody and B cell development. And with this, I'd like to thank my collaborators. The original SARS-CoV-2 antibody hunting was with Dr. Jianbin Wang from Tsinghua University, and his company recently got granted emergency use for their antibody against the disease. Dr. Qihai controlling is an expert of B cell development, and also Professor Jinyue Huang and Wang Guanbo from Peking University, who are expert of the mass spec, and also the funding source from different places. And thank you.
Thank you very much, Jianbin, for the great talk. And we do have 2.5 minutes for Q&A. So I may start first with one question. So for the long-lived plasma cells in your cluster 8, did you do some cell cycle analysis or cell proliferation and to see if the cells actually divide or they survive longer?
Yes. The cells cluster 8 can survive for a long time. We typically when we immunize the cells, and they can only survive for a very short of time. So we specifically wait until 2 months after the immunization, just to see after most of the cells just die out, and we get the cells, and they can also they persist a very long time, and also they present very high affinity of the antibody and against the cells, the antigens. So we can also confirm that using the flow cytometry. So by definition, they are long-lived plasma cells. Yes.
Okay. So another question is for the correlation between the mass spec to measure the antibody and then the BCR sequencing. So that's the correlation between protein and RNA. Like, do you expect them to be highly correlated or not?
Yeah, I believe that it depends on what kind of cell, because antibodies are from plasma cells, which are the cells excreting the antibody. So if you use the plasma cells from the peripheral blood or from the spleen, it should have, and also within the very short amount of time, for example, after the vaccination, the correlation I would expect to be very high. However, if you wait for different time point where you are using a different organ for the cellular component, the correlation may not be pretty high, because because the body is experiencing different kinds of antigen all the time. So the different groups of the plasma cells got activated on and on. Therefore, and also they all converge into the plasma, so that you can regard the plasma as a dumpster or kind of drainage to combine all the antibodies created from different body. So it depends on whether you are using the circulating plasma cells or the long-lived plasma cells or the locally activated plasma cells. So that is
thank you.
Yes.
That's great.
All right,
thank you very much, Jianbin. And we will move to our next speaker, Dr. Jinyue Liu. Jinyue is the principal investigator at the Genome Institute of Singapore. And she's going to talk about very interesting analysis of integrating scONE-seq and imaging spatial analysis. Over to you, Jinyue.
Thank you, Pan. So, hi everyone, I'm Jinyue and I'm from the Genome Institute of Singapore. So my lab is interested in looking at in decoding the human brain during health and disease using single-cell and spatial transcriptomics technologies. And so last year, I spoke at the organoids session on how we use organoids as a model system to address mental illness. And today for this technology session, I'm going to be sharing with you some of the progressive steps that we have taken and the data analysis, some data analysis solutions that we have come up with to strive towards a comprehensive use of this single-cell spatial technologies. So this audience really needs no introduction to the brain. Brain atlasing is a really exciting field, and we all know that the huge importance of this atlasing effort, and that's because functional brain wiring is going to need precise connections between very specific cellular partners as well as cell types. So here, what I'm showing you are examples of what happens when this kind of brain wiring goes awry. So on the left is an example of neurodevelopmental disorder, autism, where you get hypoconnectivity between different brain regions or hyperconnectivity. And on the right is an example of neurodegenerative disorder, Parkinson's disease, where you have specific groups of cells, namely dopaminergic neuron subset of them, that die off and leading to the symptoms that we see. Okay, so for the purpose of today's talk, I'm going to focus on spatially resolved transcriptomics. And as you can see here, there are a wide range of techniques by now, even though it's a relatively new field. And the methods that we use in our lab is something that we call multiplexed RNA imaging. And that's based on this 2015 publication by our collaborator, Hao Chen, and his co-first authors. And we have been applying these techniques to a case control paradigm where we are looking at differences in the organization of the developing brain between typical conditions versus the autistic condition. So of course, we're using brain organoids for this study. And this is really a multidisciplinary team effort because it requires different domain expertise from all kinds of people. So here's a list, and perhaps there are even names missing from it, but the list just goes on really, really long. And a lot of the analysis has been done by Lin Li, and a lot of the wet lab preparation has been done by Wan Qi. And so I'll be showing you results from various analyses done by the entire team. So before I get into those data analysis solutions, I'd just like to run over what we do once we get the spatial omics data. So here is a general pipeline for first step, really, which is just decoding the multiplex RNA imaging data. We have the raw data. We perform a series of corrections to the raw images, and that includes correcting for color channels, registration, filtering, and so on. And then we do a series of pre-processing, which includes normalizing the brightness of the bits, of the spots across the various bits, and finally decoding the spots into their gene identity. And all this is happening not just for a single tile. We have a whole series of tiles to map the entire tissue. And so the final step is really stitching them together. And when we stitch them together, what we get is tissue image of as many genes as you have probed for. So here what you're looking at is a 2 millimeter by 2 millimeter brain organoid tissue section. And we have done this for about 50 tissue sections by now. So it is actually a pretty scalable platform. And the sizes that we've been working with is something in the range of 1 to 5 millimeters, probing anything from 5,000 to 50,000 cells in each tissue section. So for our analysis, for the analysis that I'll be showing you later on, we are looking at as many as 500 plus genes. And as you can see in the correlation about RNA expression, we do get pretty good correlation with independent methods. So here's how the data looks like. So here is, I'm just showing you a single neuroepithelial bud in our organoid system. You can think of that as a unit of the developing brain. And each of these colors represent a different gene. And what you are seeing here are the location of different RNA spots for each of these genes. Here I'm showing you just 7 out of perhaps 94 genes. And you can see that just with these decoded RNA locations, we can see very fine-grained cellular organization within our organoids. And so now that we have decoded the data, we have very nice RNA spots. What do we do with them to extract biological insights? So I'll be sharing with you some of the progressive steps that we have taken towards a comprehensive understanding of our data. And here are the 5 steps or 5 different methods that we have been exploring. The first is looking at the cross comparison of spatial gene patterns. So there are several methods by now that look at spatial gene patterns. So our question really is, can we apply these methods at scale, right? Because we have a lot of data, we have a lot of cells. So, and applying methods at scale really remains to be widely tested. And so the method of choice that we have decided upon was spatial DE, and we made a few modifications to the method to enable coherent spatial gene patterns to be identified from multiple data sets to allow for cross-comparative analysis. So over here, I'm showing you the kind of data that we get. So each of these rectangles is, each of these color is a spatial pattern, and the length of the bar represents the genes, the number of genes that fall into each category from our EMFISH panel. And you can see that in each of this rectangle, you have some spots, and they are the average gene expression or the meta gene for all of the genes belonging to that spatial pattern. And what you can see is that we managed to, from 5 different data sets, we managed to describe their spatial patterns in this concise manner. And we can see the laminar organization across the neuroepithelial bud in our organoid system. So we're very happy to share that it is possible to apply these methods in a scalable way to identify coherent spatial gene patterns. So the second challenge might be, it might be an oddity of the organoid system, but I suspect that there are other model systems as well that face this challenge, which is that our cells are actually very densely packed in the organoid system. And then as a result, segmentation becomes an issue. And we do want to do segmentation if we want to do single cell analysis to get at cell type specific mechanisms. And so thankfully for us, there are deep learning methods now that have been published. And the idea here is to use image style transfer to create synthetic images that closely resemble the raw images that then can be used to train the model systems. And this approach has worked really well for us. We have managed to segment very dense nuclei, and it is as good as it gets. So where the human eye fails, the algorithm similarly fails. So it is really as good as it gets. So now that we have segmented our cells, the next step is really to assign the RNA molecules or the RNA spots in the image to each individual cell. Now, this, unfortunately for us, we did not have membrane stains. So the challenge is, how do we assign spots in the absence of visible cellular boundaries? And so we applied some creative rules that involve nucleus masking and also distances of the spots to the mask boundaries to perform our spot assignment. And the rationale being that, our cells are so tightly packed and regular in space that the likelihood of a spot belonging to a cell in our image is actually very high. And indeed, we managed to retrieve 50% of our spots, close to 50% of our spots with this combinatorial approach. And we had close to 90% of our spots assigned in each image. So now that we have single-cell spatial omic data, we can begin to we can begin to probe the spatial organization of our tissues. So there are, this field is rapidly evolving and there are new methods being invented. And here is one of them, which is spatially informed clustering, which Vipul Singhal is going to talk about later on in this session. So stay tuned. We also, in collaboration with the Bioinformatics Institute, come up with a new metric called spatial purity. So this is work done together with Lineage at BII. And the idea is to have users define the reference point. And so over here, the reference point that we have used is the progenitor neuron divide. And then we follow up with Euclidean distance transform to segment each neuroepithelial bud in our organoids, because every organoid contains multiple buds. And then we quantify the distance of every cell to our reference point. And then with this information, we are able to construct the spatial purity index, which tells us how two populations of cells are organized along the position from the reference point. And so with that, we actually did find some interesting cellular disorganization within our organoids. But with this cellular disorganization, we also predict that there are other molecular changes that happened with the spatial disorganization. And so we wanted to look at more genes. And to do that, we took the approach of integrating our sample-matched single-cell RNA-seq with the Murfish data. And the question that we are asking here is really simple. Do these two data modalities share enough information for accurate integration? And the answer is yes, for good, if we have enough genes with good data. And so in our hands, we found that with more than 200 genes, we are able to get good overlap between the two data modalities. And as you can see on the left, that's the prediction accuracy when we compare the de novo identities, cell type identities, versus the predicted labels that were transferred from the single-cell RNA-seq using mutual nearest neighbors methods. And we know that this method is reasonable because we were actually able to recapitulate many of the previously published or previously reported differential gene expression or differential pathways, such as RNA processing, cell cycle, translation, and so on. And so with that, I'll just like to summarize by saying that, through these progressive steps, we actually managed to uncover some previously inaccessible cellular phenotypes. And what we do when we are aiming for scale, we hope to be able to scale up this technology and these methods to uncover new biology for studying in neurobiology. And so with that, I'd just like to thank everyone who made this possible. Sham, Hui Kwan, Lindy, Gokhao, Nigel, Vipul, so on. Everyone, it's been quite a massive project diving in depth into this technology. And I'll take questions.
Thank you very much, Jinyue, for the great talk. And we have a time for one or two quick questions. I start first with one. So when you perform the Murfish analysis of multiple organoids, did you do some normalization to combine the data from the separate organoids?
Yeah, so that's actually a really great question. So we did spend some time looking into normalization. So I think because different imaging batches is going to give you different detection rate. And so we did try total count normalization as well as other methods by these platforms such as scTransform. And so those public methods seem to work well. And we did do, but that said, we did do very intense filtering of our data before we input them for normalization.
Thank you. Thank you very much for the great work. And for interest time, we're gonna move to the next speaker. And I like to hand over to my co-chair, Angela Wu, to chair the remaining part of this session before we go to the panel discussion.
Yeah, great. So next I will introduce a friend of mine. It's Hirofumi Shintaku. He's currently a Kubi team leader at the RIKEN cluster for pioneering research. And I think, you know, in order to give him the most amount of time to talk about his very interesting work, I'll just pass it to you, Hiro.
Thank you, Angela, for your great introduction. So I'm very happy to share our research in this meeting. So today I would like to share our technology development that allows you to explore the transcriptional noise at subcellular resolution. Right. So I believe I don't have to emphasize the importance of single-cell RNA sequencing. Single-cell RNA sequencing is a great technology. We can enjoy seeing gene expressions in millions of single cells. However, as you know, the single-cell RNA sequencing is basically endpoint measurement. You need to kill your cell sample at the time point of the measurement. So that challenges us to infer the dynamical state of single cells. Of course, many researchers have proposed various computational approaches to tease out the gene expression dynamics of single cells, including pseudo-time analysis and RNA velocity. And our group developed a unique approach that leveraged the microfluidic system, and we infer the cellular dynamics by resolving the difference in gene expression between cytoplasm and the nucleus of single cells. So today we would like to introduce you our approach, which is called the scONE-seq. So in our development, we focused on the central dogma in eukaryotic cell. RNA molecules are transcribed in the nucleus and then transported to cytoplasm, where the protein translation happens according to the expression level of RNA molecules. So in our approach, we hypothesize that around this cascade, nuclear RNA probably present the future state of the cell, while the cytoplasmic RNA does present state. So by looking at the difference between the nuclear RNA expression and the cytoplasmic RNA expression, we want to see the dynamics of single cell. To enable such analysis, we developed an approach, scONE-seq. In this approach, we first physically fractionate the cytoplasmic component against the nucleus. And then we construct two independent RNA-seq libraries to resolve the localization of RNA molecules. Today, we mostly talk about the data taken with mammalian cells, but our technique is available also for the plant cells with a cell wall, as well as the fixed cell. And we also, we today discuss about the poly-A RNA molecules, but the protocol can be easily extended to small RNA sequencing. So these papers are explaining about those extended protocols. So if you are interested in those, please check out these papers. Okay, as for physical fractionation, we use microfluidics, well, especially electrokinetics in microfluidic system, So our microfluidic system is schematically drawn, explained in this slide. So we have a hydrodynamic trap over here. So we basically capture a single cell like this. And once we capture a single cell of interest like this, we do other things by electric field. So we basically lyse the plasma membrane selectively by electric field, and then we extract the cytoplasmic component by electrophoresis. And during the extraction of cytoplasmic component, we detain the nucleus at this hydrodynamic trap. At the end of this protocol, we extract the cytoplasmic component from downstream and the nucleus from upstream. Now we have, the different subcellular components in different PCR tubes. So now I want to show you how our microfluidic system works. So what you see here is a single cell captured at hydrodynamic trap. Just by applying DC electric field, we can selectively lyse the plasma membrane. This happens within 1 second, and we extract the cytoplasmic component within 20 seconds. And after doing this cytoplasmic extraction, we correct the nucleus from this hydrodynamic trap. So by precisely controlling the magnitude of electrical field, we can keep the nucleus membrane intact, and the nucleus is still containing the nuclear RNA inside. Our approach can offer very stringent fractionation between the cytoplasmic RNA and the nuclear RNA by electrophoretic extraction. So we here benchmarked our nuclear RNA-seq data to the nuclear RNA-seq data prepped with detergent-based off-the-shelf kit. So in this right figure, we are comparing the gene expression taken with nucleus. And what we want to point out here is the mitochondrial RNA. So as you see here, the red points, mitochondrial RNA expression are high in the nucleus prepped with this detergent-based kit, meaning our approach has a smaller contamination from the cytoplasmic RNA by 10-fold. With our unique data set, we can explore the enrichment of individual genes in subcellular compartment. So we can also tell how much poly-A RNA are in the cytoplasm. In the case of K562 cells, 84% of RNAs are in the cytoplasm. The cytoplasmic RNA, which is 84% of single cell, is quite similar to that of the single cell. The nuclear RNA expression, which is only 16% of the cell, is a bit different than that of the single cell. This may be implying that the nuclear RNA may contain interesting information. Now we want to ask a question whether we can use the nuclear RNA expression as substitute of single cells. In some applications, nuclei may be useful substitutes of single cells for easy dissociation or reducing the artifact by enzymatic digestion. So using our data, we explored the correlation between the cytoplasmic gene and the nuclear gene. And by ranking the individual gene according to the coefficient of correlation, we found the correlation widely ranging from positively correlated state to the negatively correlated state. And interestingly, so these positively correlated genes and the negatively correlated genes showed very distinct biological function. And we especially got interested in looking at the negatively correlated genes. So these genes showed enriched function related to RNA splicing. So leveraging full-length sequencing of cDNA, we found the negatively correlated genes is presumably caused by intron retaining in RNA splicing genes. So indeed, by investigating the enriched function of RNA molecules with retaining intron, those RNA molecules show the enriched function related to RNA splicing. And further, we observed the degree of retaining intron and the nuclear expression of its RNA showed a positive correlation, while it didn't show any correlation in the cytoplasmic fraction. So meaning RNA splicing may make the gene expression different among the subcellular compartments. We also found that the dynamic change of cellular state may distort the correlation between the cytoplasm and the nucleus. When we perturb the epigenetic state of K562 cells, we observe the reduction of correlation around the cell differentiation. Okay, using our data, we further asked another question, whether the nuclear compartmentalization works as an amplifier or attenuator for the transcriptional noise. Eukaryotic cell separate the transcription and translation by nuclear membrane. So we just wonder how this separation acts on the regulation of gene expression across nuclear membrane. To answer to this question, we coupled our microfluidic approach with nanopore sequencing, where we basically sequence the cDNA, full-length cDNA as it is without any fragmentation. So we obtain a transcriptional noise at transcript resolution. As you see here, we observed very interesting heterogeneous diversity in the nuclear RNA, nuclear transcript. And with our data set, we found that the nuclear membrane may act as both, so amplifier and attenuator. So by doing some statistical analysis, we identified the genes with amplification and attenuation. And as we expected, in the attenuating genes, we again found the RNA splicing mediated genes. Right, so I'd like to summarize my talk. So nuclei may not be just the substitutes of single cells. They may contain their own unique information. And nuclear membrane works as signal amplifier as well as attenuator, perhaps attenuator for RNA splicing genes. And we think electrokinetics in micro and nanofluidic system may be useful, and we hope to develop further, different technologies in order to contribute to this community. And by thanking to my team member and ex-team member, I'd like to close my talk, and I'm happy to take any question. Thank you.
Great. That's a fantastic talk and excellent timing too. Thanks, Hiro. So we do have first a couple of questions from the audience. So Al asks, on one of your slides, cell cycle terms were enriched. As nuclear membrane breaks down during mitosis, what happens with the mitotic cells? Are they discarded or are they identified?
Yeah, so thank you for a great question. So even for mitotic cells, the chromosomes are probably clumped together. So mostly we can do this fractionation successfully. But in some cases, when the cell doesn't have clear, chromosome that has no association, we sometimes fail in doing fractionation. But it's really rare event in our experiments.
Got it. Bharavan asks, how do you handle the pre-mRNAs that you get from the nuclear fraction, and how do you account for that in your gene quantification?
Okay, thanks for another great question. So in our data, we basically target the poly-A RNAs. So we are not detecting the real pre-mRNA. We are currently detecting only poly-A tailed RNA, but we hope to couple another protocol which maybe enable detecting those pre-mRNA. I think those RNAs are inside the nucleus, definitely. So we hope to do another, develop another protocol.
Yeah, that's fantastic. And I actually have a question to ask you as well, since we have a minute left. So I think this is a really great system, as you've already demonstrated that it can, analyze different subcompartments of the cell which I think can be super interesting, as you've shown. Do you have any plans to do sort of more proteomic analyses from these different fractions?
Oh, yeah. So in my slide, I explained about the RNA and RNA, and we have not explored the multi-omics types of experiments. So because our, so many other people have developed the multi-omics approaches, and we wanted to develop something unique. And we found our electrophoretic approach is very powerful to provide the stringent fractionation of RNA molecules, cytoplasm versus nucleus. That's why we decided to pursue that direction. But the protocol itself is capable of doing multi-omics analysis. So once my team become bigger, probably we try multi-omics analysis as well.
Okay, very cool. All right, I think we'll have probably more questions when the discussion panel comes, but thank you, Hiro, for an interesting talk.
Thank you.
So next up we have Heather Lee. She is a biomedical scientist with a lot of experience in various different interdisciplinary fields. And she is currently a PI at the Hunter Medical Institute, Hunter Medical Research Institute in Australia. And she's done a lot of interesting multi-omic work, which I've followed since her postdoc days. So please, Heather.
Thanks very much, Angela. It's a pleasure to be speaking at this session. I'll be telling you about how we're using single-cell analysis of DNA methylation to look at the heterogeneous response of leukemia cells to epigenetic therapies. So before I begin, I just thought, since this is a technology session, that I'd remind everyone that we now have many different single-cell epigenomic techniques to look at many different components of the epigenome and even multiple epigenetic features in the same single cell. So I was fortunate enough to be involved with the development of single-cell bisulfite sequencing to look at DNA methylation across the whole genome of single cells and also related methods. So this was the first protocol that we published, and it relies on bisulfite conversion of DNA, followed by multiple rounds of random priming and strand extension. And this technique was later combined with parallel transcriptomic analysis and also analysis of chromatin accessibility in the same single cell. And it's also possible to infer copy number variants based on the read depth in the bisulfite sequencing library. So obviously these multi-omic technologies are incredibly powerful to understand how epigenetic features impact on transcription. But they are also limited by several challenges, including the throughput of bisulfite conversion chemistry and also the sequencing demand of sequencing the whole genomes of many single cells. So my group in Newcastle have also developed a much simplified approach in which we deliberately sacrifice locus-specific information from across the genome, and instead we target transposable elements using a bisulfite amplicon sequencing approach. We've called this method single-cell transposable element methylation sequencing, and a preprint is available on bioRxiv. As I said, we amplify transposable elements such as this sign-alu sequence, and these are scattered throughout our genome, and they're also heavily regulated by epigenetic mechanisms, including DNA methylation. This means that they can be used as faithful proxy measures of global changes in DNA methylation that occur in several biological contexts, including early embryonic development, reprogramming of induced pluripotent stem cells, and also in cancer. So we are able to amplify thousands of these repetitive loci across the genome with very low sequencing depth. And just to give you an idea, this is nearly 3 orders of magnitude lower sequencing depth than what's required for a genome-wide bisulfite sequencing library. And so that signifies a huge cost saving for this method. The methylation estimates that we obtain are very close to the methylation in bulk libraries prepared from matched populations of cells. And we're also able to detect heterogeneous DNA methylation induced by treatments such as decitabine, which I'll come back to in a moment. This heterogeneity is not simply a matter of picking up different parts of the genome in each individual cell, because we're able to downsample our libraries to incredibly low depth. Fewer than 5,000 reads, we still have stable estimates of DNA methylation. So our new method, single-cell TEM-seq, has a few advantages over comparable genome-wide approaches, and I'm happy to talk to this more if there's time later. But basically, we deliberately sacrifice the information in each library to substantially reduce the sequencing demand required. Because our method is compatible with parallel transcriptomics, like other bisulfite sequencing approaches, it's applicable to contexts where global changes in DNA methylation are expected and the transcriptional consequences of that epigenome reprogramming are of interest. So in our case, that's the use of hypermethylating agents in leukemia treatment. These epigenetic therapies, including azacitidine and decitabine, are cytidine analogues that are incorporated into DNA during replication. Because the 5' carbon, which is the site of endogenous DNA methylation, has been converted to a nitrogen atom, these therapies block the maintenance of DNA methylation during replication that is normally undertaken by the DNMT1 enzyme. And this leads to a passive loss of DNA methylation from the genome during treatment. These therapies are approved for use with acute myeloid leukemia and also a pre-leukemic condition called myelodysplastic syndrome. And they're also in many different clinical trials for solid cancers. Here you can see that some patients receive huge benefit from these hypermethylating agents. In this cohort of elderly AML patients, the 2-year survival rate was trebled in those patients treated with azacitidine. However, you can also see that some patients receive absolutely no benefit from this treatment. And unfortunately, despite a lot of research, we still don't have a biomarker in clinical use to tell which patients will benefit and which will not. Although the drugs are known to reduce DNA methylation across the genome, the change in methylation observed in patients is modest at 10 to 15%. And it's also similar between responding and non-responding patients. Furthermore, even the patients that do respond well to these therapies invariably go on to develop relapse. So our rationale for this study was that the response of individual cells to HMA therapy could have important implications in terms of the development of relapse or resistance. So our aim was to characterize the heterogeneous response of AML cells to HMA treatment. This is an overview of our experimental approach in which we combine treatment of AML cell lines in suspension with either decitabine or azacitidine for 3 days. And we then perform index sorting to collect single cells for multi-omic analysis. And at the same time, we seed colonies in methylcult assays to test the ability of cells to self-renew. This chart shows the level of DNA methylation in single cells in 2 batches of azacitidine treatment and in 2 batches of decitabine treatment. Some of these treated cells have as little as 20% DNA methylation in the genome, while others retain DNA methylation levels at similar to that seen in untreated cells. And this could be clinically important if these apparently non-responsive cells are then able to go on and drive relapse. We incorporate cell trace staining on day 0 of our experiment, and that allows us to correlate DNA methylation after treatment to cell divisions during treatment. The cells with low cell trace fluorescence that have undergone the most divisions during treatment also have the lowest level of DNA methylation. And this is expected given that we know these drugs are incorporated during replication. In our single-cell multi-omic analyses, we can also draw correlations between DNA methylation, chromatin accessibility, and gene expression. In these charts, I'm comparing 2 different molecular layers at a time, and each point represents a different single cell. We're also considering DNA methylation and chromatin accessibility in different regulatory contexts, so promoters, CpG islands, and different histone modifications. I don't have time to go through all the data, but you can see that there are the expected negative associations between DNA methylation and chromatin accessibility, and also that decitabine treatment has weakened these correlations. We can also look in closer detail at specific loci. So in these charts, I'm comparing two different molecular layers, and each point is a different locus in the genome. And in the first chart here, it's DNA methylation and chromatin accessibility. So we see the expected enrichment for negative associations. And we also note that many of the promoters in this particular analysis are associated with cancer testis antigens and tumor suppressor genes. However, if we look for correlations to gene expression, we have very few significant hits. And this really points to one of the limitations in these library types, because it's very difficult to have sufficient cells with coverage of a specific locus in two different very sparse libraries. However, we're continuing to collect data for this analysis and collaborating with Kim-An Lecau from the University of Melbourne to see if we can draw some biology from this data. Something else that we were really interested to explore was the upregulation of transposable elements after treatment. And that's because these drugs have been shown to act through a viral mimicry process in which methylation is lost from the promoters or from endogenous retroviral elements. And this allows the expression of double-stranded viral RNAs, which are recognized and stimulate an interferon response in the cancer cell. So we first defined which transposable elements were upregulated by treatment in bulk RNA sequencing data, and then looked at the same transposable elements in our single-cell RNA-seq data. We noted that only a subset of the treated cells were able to upregulate transposable elements, and this suggests that viral mimicry will be heterogeneous among cells, and that could have important implications for the action of these drugs. We've also been able to repeat this observation using our single-cell TEM-seq approach in other leukemia cell lines. So finally, I want to come back to this observation that some cells retain DNA methylation during treatment and ask, what's the functional consequences of this? These drugs are known to work by targeting cancer stem cells and have been shown to reduce the colony-forming capacity of cancer cells. So we performed these colony assays and picked individual colonies derived from treated or untreated cells and then performed molecular profiling. This chart shows that colonies derived from treated cells had heterogeneous levels of DNA methylation. And we also observe in the decitabine colonies that there's really a bimodal distribution here, with many more colonies having higher levels of DNA methylation than we would have expected based on our single-cell analysis. And this suggests that there's some sort of positive selection or growth advantage for these colonies. We've also performed RNA sequencing on the same colonies, and we note that despite very similar levels of DNA methylation to untreated colonies, the transcriptome is very distinct. And that shows that the majority of the transcriptional consequences of treatment are independent from the DNA methylation level. However, we can also look in more detail. This is my last slide. We can also look in closer detail to find pathways specifically upregulated in those colonies that have high levels of DNA methylation. And one pathway that came out of that analysis is cholesterol biosynthesis. So finally, I just summarized to show you that DNA methylation is heterogeneous after treatment. Some cells retain DNA methylation, and it seems that after decitabine treatment, these cells could have a growth advantage related to cholesterol biosynthesis. Thank you.
Thank you, Heather, for that wonderful talk. As always, excellent work doing multi-omics, including the methylation work. So maybe a question from me first. On the methylation side, I'm just curious, what are some current bottlenecks that you're still facing? I mean, your method is obviously fantastic, and it's a great improvement upon some of the previous ones. Are there any continuing challenges that you still face?
So bisulfite conversion chemistry requires cleanup of DNA after treatment. So we're using magnetic beads for that. So one possibility that might overcome that limitation is if single cells can be encapsulated in agarose beads or some other media in a droplet encapsulation technology that would allow us to do the bisulfite conversion in bulk on many cells in one tube.
Mm-hmm, mm-hmm. Have you attempted any of those enzyme-based type of methods in your pipeline, or the bisulfite is comparable to that in terms of what you just described?
Right. So there are new methods like the EM-seq technology that replace bisulfite conversion with enzymatic activity. I haven't tried them. They will depend on the efficiency of the enzymes, and yeah, it could be very interesting to see if it's compatible with indexing before enzymatic treatment.
Yeah, yeah. And then sort of to follow up on your very last result, which seemed very exciting. I mean, do you have any follow-up studies, maybe more kind of preclinical related work based on that finding?
No, that's very new. I did have a quick look at the literature, and it does seem that cholesterol biosynthesis is important for helping cancer cells to protect themselves from drug-induced cytotoxicity. So it seems to make sense, and we will follow up with co-treatment assays to check that it's real.
That's great. Actually, so we got a last-minute question coming in. Maybe you could respond to it in the chat box, since we ran out of time for the talking part. Thank you, and we'll see you back in the panel discussion. Okay, so next we have up Vipul Singhal, and he's a postdoctoral fellow in Dr. Sham Pavarkar's lab, and also the lab of Dr. Hao Chen at the Genome Institute of Singapore. So I'll hand it over to Vipul. I think you might still be mute, Vipul.
Oh, sorry. Hi. So
yeah, you're good now.
Thanks for the introduction. Hey, so today I'll be talking about a spatial clustering algorithm that we've developed. It's called Banksy, or Building Aggregates with a Neighborhood Kernel and a Spatial Yardstick. So nobody needs an introduction to what clustering is. Basically, the idea is if your cells are similar in gene expression, so they sit close to each other in gene expression space, we tend to cluster them together, and if they share certain markers, or if they express the current markers, we call them a single cell type. Question is, spatial technologies are coming up, and can we do something? Can we use the spatial information somehow to improve clustering? Right now, what most of us are doing is we're just using non-spatial clustering, what you might find, like graph-based clustering you might find in a pipeline like Seurat to do clustering with. So the question is there some natural way to incorporate spatial information to perhaps improve the sensitivity or the accuracy of clustering? And the answer is, well, yes, you can. One simple thing you could do is just try to append geometric coordinates, the XY coordinates of a cell, to the gene expression vector of the cell. This is quite naive and doesn't actually work. To see that, consider what happens if you look at brain organoid buds, the way we see in Jinyue's presentation. So suppose I have two buds, and I've got two layers in each bud, the layer with blue cells and green cells, and the same in the other bud. And so the blue cells will sit in the same location in gene expression space. So this axis is the gene expression space collapsed to a single axis, and this is geometric locations or physical space locations collapsed to a single axis. The blue cells will all sit in the same location in in gene expression space, but different locations in geometric space, right? So, and the same thing for the green cells. They're also in different locations in geometric space. So they show up as different clusters. So here we would just end up with 4 different clusters, where really there are only 2 cell types. So global coordinates aren't the right thing to include as spatial information. And we can try this on actual real data, and we find that with non-spatial clustering of, so this data is from Jinyue, with non-spatial clustering, you reproduce the bud structure. Whereas when you put this, when you append the geometric coordinates to your vector, cells that are far apart that really should be the same cell type, they get called out as 2 different cell types. And that's just because they're very different in their geometric coordinates. And the situation gets worse as you weight the XY locations more and more. So this isn't quite the right way to do it. A better way to do to incorporate spatial information is to regularize things locally. What I mean by that is to use the microenvironment signature of a cell, so a cell's neighbors, and take their average expression and append that to the expression vector of a cell. So now a cell's feature vector is a hybrid feature vector of its own expression and the average expression of its immediate physical space neighbors, right? And in terms of dissimilarity matrices, what this is saying is the dissimilarity matrix in this hybrid space is basically a convex combination, or just a sum of the dissimilarity matrix in gene expression space and the dissimilarity matrix in neighbor expression space. And so going back to the thought experiment on brain organoids, we see that the blue cells here, they'll tend to get clustered into the same group because they're similar. So the blue cells here versus the blue cells here, they're similar in their own expression and they're similar in their neighborhood expression. So both groups A and C are similar in both own and neighbor expression. Same thing for the green cells. So you might ask, okay, well, what's the utility of this? I'll get into that with some examples, with some case studies. But before we do that, I want to touch very quickly on what this lambda parameter is, because it's quite important. So the lambda parameter is essentially a weighting parameter. You're just saying, how much importance do I want to give to a cell's own expression vector versus the average expression of its neighbors? The larger lambda is, the more weight I give to a cell's neighborhood expression. And so at lambda equals 0, you basically have your usual graph-based Seurat non-spatial clustering. So you're only looking at own expression. And at intermediate values of lambda, you go into this hybrid space, this product space of the own expression and the average neighborhood expression. And here, a single cell type can get stratified into subgroups by neighborhood expressions. And there is utility to this, as we'll see in a bit. And finally, as lambda goes to 1, you basically collapse out the own expression space, so that doesn't exist anymore. And what you have is all the cells sit on what the average expression of their neighbors is. And so what happens here is, if you cluster cells only on their neighborhood expressions, you get a zone finding method or a domain finding method. So that has utility as well for different different use cases. Okay, so looking at some data sets, the first one, the famous mouse hypothalamus data from Jeff Moffitt's lab, they have this, the mature oligodendrocyte cluster, for example. And the yellow cluster here is showing you what happens when you use non-spatial clustering. So just looking at the own expression of cells, and you get all the correct mature oligodendrocytes. But if you incorporate neighborhood signatures as well, you get a splitting of this cluster into 2 subclusters, a denser one and a sparser one. And if you look at just the own expression signatures of the marker genes of mature oligodendrocytes, you find that there's a barely perceptible difference between. So there's really not much difference in the own expression signature of these 2 subgroups. But looking at the neighborhood expression, it becomes clear what happened here. The sparse subcluster doesn't really have in the neighborhood of the sparse subcluster cells, the markers for mature oligodendrocytes aren't strongly expressed. Whereas for the dense subcluster, the oligodendrocyte markers do show up. And so then we might say, okay, well, if we just rescale the colors a little bit, we can actually see that there are genes with subtle differences between these 2 subclusters. So there are genes that are higher expressed in the sparse subcluster, and there are genes that are more highly expressed in the dense ones. And the same thing happens with excitatory neurons. So again, non-spatial clustering gives you a cluster of excitatory neurons. And with spatial clustering, using the microenvironment signature, you can split things up, and you have some subtle differences which are very obvious in this heat map because we've scaled things to make the differences more pronounced, but they're not pronounced enough for non-spatial clustering to tease them apart. The next thing I want to touch on is, so the take home, by the way, for the previous slide was basically subtle differences, subtle transcriptional differences might be easier to tease out if those differences also correlate with differences in microenvironments. So if cells are in different microenvironments and if they have different subtle differences in their expression, then we can tease them out. For the next thing I want to show you guys is a denoising aspect of this algorithm. So looking at the slide-seq data set where you have these bar-coded beads and you put your tissue on top of that, where multiple cells can sit on a single bead and contaminate the expression measured in a bead, multiple cell types can contaminate it. We find that, well, Banksy is able to help denoise this a little bit. So recently there was a study, RCTD, robust cell type decomposition, where they showed that with the slide-seq data, often you call out granule cells in non-granular layers. And this isn't quite right. And so we asked, could we improve this situation where we use the microenvironment signature somehow to denoise these cells? And we find that yes, well, non-spatial clustering, just like the RCTD paper reported, you call out a bunch of granule cells in the non-granular layers. And with Banksy, in an unsupervised way, we're able to denoise the clustering of the granular cells. And this agrees with what the RCTD paper, what that paper shows as well. And slide-seq version 2, that's a less noisy data set, but still there are some cells that get misclassified, and RCTD improves the situation in a supervised manner. So they are using a single cell reference data set to help improve the clustering. Banksy is just leveraging the spatial information. So in an unsupervised way, it's able to get the clustering done. And the same thing holds for molecular layer interneurons and so on. So this is useful because you might not always have a reference single cell data set to use something like RCTD with. So we thought that a unsupervised method is also valuable here. And finally, I want to touch on the zone finding aspect. So what happens when lambda is very large? So when lambda is very large, the own expression signature collapses. And what you have is all the cells with different expression signatures, they all get clustered by the average neighborhood signature. And so we can use this to, for example, cluster out zones. So here in brain organoid data, we see that there's this middle necrotic core area. And this area has a bunch of cells of low quality with a particular signature, and clustering doesn't work to remove these cells for pre-processing purposes because you would remove a bunch of cells in the area that you do want, where the neuronal layers are, where the buds are. But in zone finding mode, Banksy is able to very, very precisely sort of capture the necrotic core and some misdifferentiated cells. And so we can remove these as a pre-processing step and then focus our analysis on the part of the tissue that we do want to look at. So it's sort of like a region segmentation sort of method. Okay, so I'll end with a quick summary here. So cell typing, basically Banksy is able to do cell typing, and the take home is that a cell is known by the company it keeps, or microenvironment signature is the way that spatial information mediates its influence on a cell. So that is the thing that you want to leverage. And it's fast and scalable. So one thing I like about this method is that it isn't inventing a new, it's sort of leveraging existing fast clustering pipelines. So for example, graph-based Leiden or Louvain clustering are state-of-the-art. They scale to millions of cells. So we wanted to reduce the spatial clustering to that problem or map it to that problem. And so we did that. And so the next thing is that it's sensitive, so it's able to find subtle differences in expression of cells, broadly applicable, right? So works on proteomics data, can work on, and here we showed fish data and slide-seq sequencing-based data, but there's no reason it cannot work on proteomic type data sets. And finally, it's multifunctional, clustering as well. So I'm going to end with thanking my co-first author here, Nigel Cho, and Joseph Lee, who's an intern in our lab, And Sham, Kakao, Huikwan, they're the PIs on this, and Jinyue provided data and others. Okay, thank you very much. I'll take questions now.
Okay, thank you, Vipul. Very nice talk. We have a question from the audience. Martin asks, what clustering algorithm are you using, and how does the selection of different parameters, for example, number of neighbors, affect the clustering results?
Yeah, so great question. So we're right now using lead-in clustering, but we've played with Louvain clustering. You can really use any clustering method. Like if you wanted to, you could use hierarchical clustering. That wouldn't scale to more than a few thousand cells, but you could if you wanted to. So what we're doing is we're defining a new coordinate system. And once you've transformed your problem into this coordinate system, you can feed it into any clustering method that takes a feature cell matrix or feature object matrix. Right? So it's not even really restricted to biology per se, right? If you have spatial data in geology, you can use it there as well. There's nothing here that says it has to be in biology. As for parameters, very good question. So so there's what we call K expression, which is the number of expression neighbors in expression space. There we find really any number works. We've tried all the way from 25, 30, all the way to 100. Doesn't really matter. It's fairly robust. If you have a larger K, you get fewer clusters. So if you find that, oh, you're merging clusters, just reduce the K a little bit. The number of geometric neighbors, again, that doesn't matter, because what we're doing is we're actually taking a weighted average, where the further you are from your index cell, the less contribution you give it. And so the kernel function there, we've tried Gaussian kernels, we've tried 1/r type kernels, we've tried 1/r. It doesn't really matter. Again, it's very robust to these differences. So we've just, in the package that we'll be releasing soon, we've got some sensible defaults, but you can try the various other options, but you'll find your results qualitatively remain the same. So
there is a question about whether there is a publication that we can check out.
Yeah, so that's coming soon. I'm hoping in the next few weeks. So we'll be releasing all of that. So keep an eye out for it.
That's great. I have Quan asks, does the use of neighbor averaging mask specific signals of rare cells within their abundant neighbor cells?
Yeah, so it's a great question. So what will happen is, if a cell is very strongly of a different cell type, right, and it's very different from its neighbors, then it will not get masked and be made into the same cell type as its neighbors, as its neighbors. But if it's on the boundary, if its transcriptional signature is very similar to its neighbors, but could be some other cell type as well, then it will tend to get labeled as what its neighbors are. So it's the noisy cell types that you aren't sure of, those get relabeled. But we do find that there are many cells in a different neighborhood, not of the same kind, they stay as their own cell type. And this is a matter of staying within a range of lambda values, which we provide. So, that intermediate range from 0.15 to 0.3, there you're fairly safe. So, yeah.
Okay, great. So in the interest of time, we're going to move on to our last talk of the session, which is from Professor Longqi Liu. So Longqi is a chief scientist of single-cell omics at BGI Research, and I believe he's going to talk to us about their own spatial omics technology today. So very exciting. I'll just hand it over to Longqi.
Okay. Thanks, Angela, for the nice introduction. So first of all, I would like to thank the organizers for inviting me here to give a talk in Human Cell Atlas Asia meeting. So today, I'm very excited to introduce a new technology on spatial transcriptomics. So first, I think I don't need to emphasize again how important the single-cell sequencing is. But one point I really want to mention is that cells are actually in a specific spatial context. Yeah, but single-cell sequencing actually lose context, which means that you can hard know where the cells from and what the neighbor cells surrounding each cell types, and whether there's cell-cell communications, for each in specific microenvironments. And so a critical need in this field is the development of spatially reserved technology. The good news is that actually great efforts have been put in the development of spatially reserved transcriptomic technologies. And a general used strategy is actually based on in situ hybridization, which allows the profiling of a set of genes. And the emergence of recently developed chip-based technologies such as SLICE-seq and Vision can allow the profiling of genome-wide gene expression with a relatively high resolution. And in terms of chip-based spatial transcriptomic technology, actually there's a bottlenecks in currently available technologies. And the limitations include the resolution, which is related to the size of spot and the distance between spots, and also the chip size, which decide the size of organs that can be used for that this technology can be applied to. So today I want to introduce a new platform that can address these challenges. And the idea comes from the DNA nanoball sequencer developed by BGI team. And this sequencer is for high throughput DNA sequencing. And actually in this sequencer, we are using our very high density DNA nanoball pattern array. And in the DNA nanoball pattern array, you can actually load billions of DNA nanoballs onto the chip. And each DNA nanoball is around 220 nanometer. And the distance between DNA nanoballs is around 500 nanometer. And after sequencing of the DNA nanoballs, you can actually first get the sequence of each DNA nanoball and also get the coordinate of the DNA nanoball. And this platform inspired us to the development of ultra high throughput, ultra high resolution, spatially reserved technology, which we termed scONE-seq. And this is the entire workflow. So basically, we first synthesize a library of short DNA fragments, which contains a random sequencing as unique barcode for each fragment. And after rolling cycle amplification, you can get billions of DNA nanoballs, and you after sequencing of these nanoballs, you actually get the second strand of the DNA nanoball, and of course with spatial information. And then we hybridize the nanoball with poly-T and UMI containing fragments. And by this way, we generated the spatially barcoded scONE-seq chip. And afterward, we have put the tissue sections onto the slide, and then we did in situ MI capture and the library construction. And in the second round of sequencing, you actually get the sequence of mRNA and also the coordinate of each DNA nanoball. And by integration of first round and second round sequencing information, you can actually reconstruct the spatially reserved transcriptomics map of the tissue. So far we have different type of different size of chips for different type of organs. And the largest chip is over 170 square centimeter, which means that you can actually provide very large tissues with scONE-seq. And this is an example of the non-human primate brain, which can be perfectly covered by our chip. And this is direct comparison between scONE-seq and the other available chip-based spatial transcriptomic technologies. And in terms of MR capture efficiency, you can see here, actually scONE-seq can capture quite high number of MI transcripts when comparing to other high-resolution spatial technologies. And also you can see highly consistent gene expression pattern between scONE-seq and the in situ hybridization methods. So when you zoom into specific regions, you can actually see the MI transcript aggregates. When you compare the sequencing data with imaging data, which shows the nucleic acid locations, you can actually see perfect match between the sequencing data and the imaging data. This indicates that actually scONE-seq can achieve cellular resolution. So next, I would like to give an example to showcase the power of scONE-seq in dissecting tissue complexity. As we know, the embryogenesis is probably one of the most fascinating questions in biology. That previous studies has been mainly focused on earlier embryonic time point because only few cells are present. However, the later stages, particularly the stages after gastrulation, has been poorly understood. So now with scONE-seq, we are able to profile the the gene expression at whole embryo mount, at whole mount embryo level. And so what's the data look like? We applied the scONE-seq to profile to generate the first high definition spatial temporal transcriptomic atlas of mouse organogenesis. And here you can see the spatially constrained clustering showing very clear pattern of each organs. And when you zoom into different organs, you can see the microstructure of different organs. As you can see here, heart, the limb, different layers of spinal cord, and different layers of adrenal gland. So as we know, the transcription factors actually play a very important role in the cell transition during development. But this has been well studied with single cell sequencing. But what does it look like when you consider the spatial information? So then we applied scONE-seq, which is an algorithm that can calculate transcription factor activity and has been very widely used in single cell sequencing. And by applying scONE-seq to our scONE-seq data, we identified a lot of transcription factors that show specific enrichment in different parts of the embryos. Like shown here, the example GAT4 in the heart, SOX2 in the brain, and FOXA2 in the node code. And if you look into the specific regions, for example, if we look into the brain part of the E9.5 embryo, you can actually see more clusters when using the regular activity score. You can see 5 more clusters. Although all these brain are SOX2 positive because all of them are neuron progenitor cells, but you can also see specific transcription factors that regulate different parts of the brain, and these are also corresponding to the no brain regions such as telencephalon, diencephalon, midbrain, hindbrain. And when you look into their brain across all stages and with higher resolution spatial clustering, you can actually get different parts of the clusters of the brain. And with the regular activity score, you can actually identify the transcription factors underlying the development of different parts of the brain, such as diencephalon, the pallium. And in addition to intrinsic transcriptional regulation, actually the extrinsic factor also play a critical role during regional specification. So next, we also investigated the morphogens that potentially regulate the different parts of the brain. And interestingly, we identified a lot of morphogens located in the secondary organizers. Like shown here, the Wnt factors are located in the roof plate of the brain, and the sonic hedgehog families, located in the floor plate. And you can also see the FGF family factors located in DLI and ISO secondary organizers. So this means that with the same scONE-seq profile, you can actually simultaneously identify the transcription factors and the morphogens with the same section. So another interesting event is that actually during the development, the neurons are migrating all the time. And what does it look like? So as we use the E15.5 brain as a principle of concept to show this case. And you can see here, this is a high resolution clustering of E15.5 telencephalon. And you can see here different layers of neocortex and different regions in the subpallium. And interestingly, you can see the layers including ventricular zone, subventricular zone, intermediate zone, and the marginal zone. Interestingly, in the cortical plate, you can also see heterogeneity along the rostral to condor direction. And to study the neuron migration, we use the arm velocity to the scONE-seq data, and we interestingly reconstructed 2 different migration pathways. One is from the ventricular zone to the cortical plate. Another is from the MG to the cortex region, and this reflects the migration of interneurons, which shown here, the tangential migration and the radial migration of projection neuron from the neuron radial glial cells. And so how about other cell types? So we also mapped by using the single cell markers. We also mapped all different cell types at spatial resolution. Here you can see all different cell types, the dynamic spatial localization across all cell types, including interneuron, the neuron progenitors, and the projection neurons. So in summary, so by using the mouse organogenesis as proof of concept, we can show that scONE-seq has great potential in dissecting organ structure, cell microenvironment, and cell migration at a centimeter scale. So in the end, I also want to highlight several manuscripts, 6 manuscripts that we posted to by archive. These are great efforts from my colleagues and collaborators. And this manuscript showcased the power of scONE-seq in dissecting tissues from different type of tissues and different species. So this is the end of my talk. I want to thank my colleagues from the ST-Omics team from BGI for the joint effort developing scONE-seq technology, and our collaborators from different institutes for the data analysis and the interpretation. Thanks for the organizers, and I'm happy to take any questions.
Thank you, Wang-Chi, for a very interesting talk and really nice data that you've shown there. So no questions from the attendees yet, but I do have a couple. So first of all, I'm curious about, you mentioned in the very last slide something about resolution, and I may have missed it earlier. So what is the resolution of this method?
Actually, this technology is based on the nanoball, and each nanoball is actually 220 nanometer in diameter, and the distance between different nanoball is 500 nanometer, which means that each cell contains around 400 nanoball. So the resolution is actually very high. You can possibly achieve subcellular resolution. So actually, in some of our data, we can see the separation of exonic rates and intronic rates, which means that you can actually get even the location of nuclei and the cytoplasm. Yeah.
Oh, that's really nice. And also in your images of the brain, I mean, you show like different zonation that you could identify. So I myself, I mean, we've been talking about the Atlas project that we've been working on, and I analyzed some of the sperm data, the spermatogenesis. So I mean, in the embryos and so on and so forth, rather than sort of more distinct boundaries of different tissue types, when you have a more continuous gradient like spermatogenesis, where spatially is also continuous, is that also observable on your sequencing spatial-seq data?
I think so. Actually, this is the advantage of our scONE-seq. Actually, in the brain, like I show with the neural migration, you can actually, because the signals is very continuous, you can actually see the migration or even the differentiation paths of different cell types. So I think would be very interesting to explore different type of cells in the continuous patterns of different type of cells at spatial resolution. Yeah.
Mm. And what about like trying to construct almost like a 3D model? I mean, you guys can do such a large section that theoretically it may be possible to stack all the slices from one embryo.
Yeah, actually, right now we are doing this. We have completed actually 3D atlas of monkey brain, which is really large. It's around 5 centimeter by 3 centimeter. And we actually had over 100 slides for the monkey brain, and wow. We already did 3D. And I think this paper will be coming out very soon. But it's really interesting. If you look at the 3D atlas, you can see difference actually across different regions.
Oh, that's great. Very quick question. Do you segment the cells or bin the signal?
Yeah, segment. Yeah, this is very great question. cell segmentation is very important for this technology. Actually, we have several approaches for cell segmentation. And the first is that I have shown the image of the nucleic acid. Actually, with this image, you can do image-dependent cell segmentation because the image is from the same section. And the second approach is actually, since from some of the sections, you can see the, with the scONE-seq data, you can see the risk aggregates on the spatial at the slides. By the aggregated rates, you can actually do cell segmentation by image-free methods. And of course, actually, we can also see the variation across different types of tissues. For some tissues, you can hardly do cell segmentation, but alternative ways that you can actually, using single cell sequencing data, you can project the single cell markers into the scONE-seq data, and by this way, you can also see the cell types.
Okay, great. I think we've run out of time for the talk section. So next we move into the panel discussion.